Hi, thank you everyone. I'm sorry, I'm just getting the last of my lunch off my lips here, so I'll try not to spit up too much sandwich on anyone or anything like that. But I really want to thank everyone for coming back and joining us this afternoon. It's a really interesting session. I'm looking forward to this today. We're going to have two presentations. It's going to be quite interesting, and then we're going to have a little mini debate afterwards, or a little Q&A session. So there will be a quiz afterwards. We expect you all to pay attention and come up with some good questions and make it as difficult on all of us as possible. But very quickly, open data has been a very hot topic recently. Obviously, with the technological advancements that have been occurring recently, especially around AI, the value of large data sets has become something that's been very critical to drug discovery, drug development, particularly inside clinical trials as well. This has been all very, very important and very, very useful data. Large pharma and large biotech companies and small biotech companies have been very intransigent about sharing this kind of data with the public. It's kind of considered some of their core gold, their core IP, their core know-how, and a lot of effort and time is put into, and money is put into collecting, curating, annotating, and retaining that data. So this is actually going to be, I think, an interesting chat because there are other ways. I mean, a large part of this whole symposium is about looking at drug discovery, drug development in different lights, in different ways, and how to do this. So today, we have two speakers who are looking at this problem of accessing and sharing large amounts of data in a different way, in an open science way. And so I think it's going to be quite interesting to hear what they say, how they've been going about this process. So each of them are going to get about 20 minutes to chat. There'll be a very short Q&A after each one chats in case there's some hot topic you want to get off your chest right away. But after that, we're going to have just, as I said, a mini panel session, and we'll go from there. So without further ado, I'd like to introduce the first speaker. He's allowed me to call him Ben, but Benjamin Haibe-Kains. How did I do? I got pretty good with that. Yeah, thank you. So quite a list here of accomplishments. He's the head of data science at the Structural Genomics Consortium. He's a senior scientist at the Princess Margaret Cancer Center. He's an associate professor at the Medical Biophysicals Department of UT, and he's going to speak with us about AIRCHECK, the Artificial Intelligence Ready Chemical Knowledge. So I'll pass it over to you, Ben. Thank you very much.
Thank you very much, Owen. You have to be a little bit kind with me. I'm absolutely not a chemist. I don't really know what I am anymore, but probably more of a data scientist / AI lover. But I really care a lot about data. So when the Structural Genomics Consortium approached me to see whether I could build a platform to really share data at scale, I was very excited, especially because of the open science mission of SGC. So I'm going to present AIRCHECK. It's our new platform to share very large-scale chemical screening data. And you're more expert than me on this topic. I'm still very naive about many of those concepts, but you can see that the hit discovery pipeline is a multi-step process. But along that process, there are a lot of opportunities to basically generate very, very big data and then use machine learning and AI models to improve the hit discovery, the identification of hit. And what's really, if you look at the AlphaFold story, is really, really telling. It's basically a 20-plus year journey in collecting, annotating, creating data, large amounts of data in a very systematic way, organizing open competition to basically challenge the community on an equal foot, so very unbiased assessment of the performance. And finally, you get breakthrough, and we also the AlphaFold breakthrough that was awarded the Nobel Prize. The prediction of protein structure has really kind of improved in terms of performance dramatically because we had so much data. So it's really at the source of many AI discoveries, many AI breakthroughs. There was also a very high quality data set behind the scene that made it happen. And the question now is, can we have a similar success story for hit discovery? So when we thought about building a platform to share data, we wanted to accomplish two main objectives. The first one is really to enable, facilitate the development of AI methods in a completely open way with really transparency and reproducibility in mind, so that people could analyze and share data on a large scale. And we want also to increase collaboration within the community. So we want to make sure that those AI-powered drug discovery pipelines are really accessible to all researchers all over the world, and build strong networks. So I think you've heard from Albert yesterday the creation of the mainframe network, and that's obviously a key network to leverage the data we are going to put on AIRCHECK. But there are still many challenges when it comes to aggregating data to enable AI in hit discovery. The first one is that we need technologies that are fairly consistent. We cannot use bespoke assays that are tuned for every single protein. We need to have technologies where we can run the same assay again and again for all those target proteins, and so that we can have this rich, highly structured data. Then we need to aggregate those data. Obviously, AI really benefits from getting very large data sets. So how do we consolidate all those data so that and make it really easy for machine learners and AI experts to analyze the data? Then models always generate predictions. That's a fact. Some are really bad, some may be good. So you really need to spend a lot of time to validate those predictions, and you have to do it at scale. And then finally, in terms of benchmarking, you can really, and we've seen it with AlphaFold, you really need to set up the benchmark so that the community can make sure that the community can assess the progression in the field in a very unbiased, systematic manner. When I read a paper claiming they can do, I don't know, a 90% success rate on whatever, I never trust them because I've been there. It always works in your hands, and when somebody reproduces your results or your model, it always falls pretty, the performance is very down. So you always need to have a bit of a neutral broker when it comes to do unbiased benchmarking. And so we start thinking about how we can actually try to address all those challenges. And what we call the AIRCHECK process, the first one is to choose technologies where we can do efficient screening of chemical compounds. The second is to find a way to aggregate those data at scale. Then we want to make sure that we are building AI models that are very transparent, reproducible, and we can manage those models, generate predictions, and validate those predictions. And so the first step is we basically choose two main technologies, and I think you already were exposed to those technologies in the previous talk. So the first one is DNA encoded libraries. So this technology is able to basically screen billions of compounds, and you attach a DNA tag and you sequence to make sure that to detect the compounds that stick to the protein. And the beauty with that approach is that you can really have a super deep exploration of the chemical space. It's fairly cost effective. The turnaround time is pretty low. However, those data are very, very noisy. The DNA tag may actually interfere with the binding of the compound to the target proteins. And you get, obviously, a very massive class imbalance. You get a few positive, few binders, and literally billions of non-binders. The second technology that we selected, at least for this first version of AIRCHECK, is a technology developed by Yui Peng at University and Levin at the University of Toronto and SGC. It's basically a mass spectrometry method where they use the enantiomer as kind of positive control to make sure to kind of increase the number of true positives. The beauty with that approach is that it's label-free, so you don't have to add a tag. It's very high sensitivity, and the screening is fairly rapid as well. However, it's fairly small scale. You can do maybe 500,000 compound screening compounds that way. You need pretty expensive instrument, and you still have class imbalance, but that's kind of the nature of the problem. So using DEL and ASMS, we want to generate very large-scale data, but we also want to prepare those data in a way that it's very, very easy to do AI modeling on them. I was raised a bioinformatician and computational biologist. I know for a fact that I spent 80% of my time just wrangling with data, and that's no fun. You really want to focus on the machine learning and the AI aspect, and not so much about organizing data. And so that's why in AIRCHECK we put a lot of emphasis in organizing the data in a way that they are kind of analysis ready or AI ready. So for that, you need to have a very good data dictionary. You need to be able to define your formats very clearly. You want to make sure that the way you process the data is completely transparent and reproducible so that if you make an error, you can fix it. If there's a new pipeline, you can rerun it or new data. And we also want to bring the code to the data. A data on its own, a data set on its own is very interesting, but if you have a piece of data that shows how you could use it, it's kind of a manual for the data, you really increase the value of it. And we want to be, obviously, we want to be machine readable so that we can do large-scale AI and machine learning, but we also want to be human-friendly. And so we are going to basically provide Pocket files. Those are file optimized for large-scale data set and can be used for AI, but we're also going to generate spreadsheets so that you can open it in Excel, my least favorite program of all time, to basically look inside and see what the data look like. And we kind of partner with Google to see whether we could generate process those data at scale and store them in a database. And as you can see here, and I won't go into all the details, but we use containers. So containers are kind of virtual machines where you define everything, all your software libraries, your type of architecture. We use pipeline orchestration called Apache Airflow so that all the code is kind of running in sync and we can easily reproduce. We use BigQuery as a large database for the Dell and ESMS data. And at the end of the day, we generate those files that we sometimes call data packages that are really easy to use for AI. And Google Cloud is really enabled this to do this at very, very large scale. And so the data on AIRCHECK is actually growing pretty fast. You can see here we have 3 sources of DNA encoded library, one from 2 companies, XCOM and HitGen, one from an academic partner, Baylor College of Medicine. And SGC is really taking on itself to kind of generate the first generation of ESMS data. And you can see that we have, if you don't count the library definition for Dell, which could be terabytes, hundreds of terabytes of data, if you just look at the positive and negative, the binders and non-binders, you can see that we have quite a few gigabytes of data already with a lot of ESMS data being generated recently. So now let's talk about the AI models, because at the end of the day, we want to prepare the data so that we can do ML and AI for hit discovery. And we want AIRCHECK wants to, or at least we want AIRCHECK to support two main paradigms. The one that you probably all know is called data to model. So you download the data from the platform and you do all the modeling on your side. You may have your own computers, you may want to run your own kind of closed source internal algorithm on the data. And we definitely support that. But we also want to support a paradigm of model to data. So let's say you don't have your own infrastructure. Let's say you're from a low-income country. We want people with just a laptop and an internet connection to be able to do the same quality of AI modeling than anybody else. And that's basically where you can use Google Cloud to do all the AI modeling. And there are some barriers to do this, but I'll explain later on how we want to overcome those. Okay, so here's a typical ML AI pipeline for hit discovery. It's very naive. There are many, many different ways to do this. But you basically start with those very rich Dell and ESMS data. So you have a series of binders, non-binders, so positive and negative. You can basically compute any features that you want on those chemical compounds. Let's say you compute a series of fingerprints, for instance. Then you do your training test set to make sure that you're not overfitting when you train your model. Then you kind of tune that model with the data. And once you have the model, what you do is you take a chemical library of drug-like compounds, such as enamine, real, and then you run your model on those, and you basically let the model decide which drug-like molecule is more likely to be an efficient binder, and then you do the experimental validation. Courtesy of James Wilnitz and Alexander Tropsha at UNC, they basically built the first end-to-end open source model for hit discovery. It's on ChemArchive. And really the idea is that there is no chemist involved. The model can really do a hit discovery from the fingerprint, from the DNA, and could elaborate data up to generate predictions for for all drug-like libraries. And it's basically using three sources of data, three types of fingerprints to do those predictions. We care a lot about making sure that those models run reproducibly on the platform. So we use something called MLOps, machine learning operations, to ensure that we know where the model is, we store all the prediction, we version those models, that all the software environment is properly documented, so that it's very easy for people to basically take the model, modify it, or replace it with something else. So in terms of validation and prediction, I'm not even going to pretend I know those technologies, but we use three kind of bespoke assays for the experimental validation. So surface plasmon resonance, SPR, dynamic light scattering, DLS, and weak affinity chromatography. So with those assays, we usually test systematically a dozen hits from our best models, so that we always have a little bit of validation for each data set. But when we are going to organize open competition, and you've heard Mathieu Shapira and Albert Antonin yesterday, we are going to organize those competitions and test a lot more of those models' predictions. So AIRCHECK is really also a platform to build a community. As I said, we want more people to collaborate. And so here, the beauty with Google Cloud is that it's accessible from everywhere. And as I said, we want a very low bar of entry. If you just have a laptop, a Chromebook, let's say, and an internet connection, you can literally go on the platform and do very sophisticated AI modeling without having to move the data whatsoever. We want a platform that can really scale up in terms of data, but also in terms of models and a number of users. And we want the access to be equitable, and we want people to collaborate as much as possible. So here's the thing about the cloud. It's a bit of a trap. You put your data there, and then you have to pay every time you get your data out, and you have to pay for all the software or services that you use on the platform. But it's also super convenient. So what we try to do now is to work with cloud providers such as Google to provide free research credits for people from low-income countries. So if you come from a country for low income, you're basically going to be able to use Google Cloud for free for your AI modeling. That's the hope. And we're also partnering with other organizations such as Computing for Humanity, so that we duplicate the data and so people can use other kind of high-performance computing for free. So I'm not going to present that, but we're very excited to work with the mainframe network to really get as many chemists, cheminformaticians, machine learning, AI experts to look at the AIRCHECK data and potentially tell us what we did wrong and do better in the future. Okay, so in terms of roadmap, we're really at the beginning. I think we had a prototype early this year. We released a new version a month ago, and we're still integrating more and more ML AI models, more data sets. We also want to make the platform more connected to other platforms, so there will be more kind of interfaces to play with it. And then the real test, I think, is going to be organizing those large open competitions where a lot of people connect, use the data, do the AI modeling, and really see if the platform is robust. So we've teamed up with a lot of partners. You can see here, I'm very happy to work with Conscience on this. And so here I'm going to show you like a few screenshots of what AIRCHECK actually looks like. So you get the landing page here, where you basically, if you scroll down, you have a series of information about what is AIRCHECK, what's the mission, what kind of data is in there. If you look at specific data, you can click on data sets, and then you see all the DEL and the ESMS data. You can download some data right away with a very simple click and checkbox saying that you're not going to do anything stupid with the data, like building biological weapons. For some other data set, the access is a little bit more tricky because we work with companies and they want to make sure you're not reverse engineering or their library and building the smiles back. So you have to basically apply, have a short project description, and the company will basically grant you access. We also have all the data dictionaries, so you understand exactly how those data are formatted. Here's an example with ESMS. And we actually also provide version history. We already had a case where the data were badly processed, and so we had to push a new version of the data. And we not hide that fact. We have all the versions of the data with explanation about which version what is attached to a version. And if we improve the way we process the data, we're going to release a new version. So if you look at the number of targeted proteins we already studied, so for DEL, we had around, let me see, 14 proteins. For ESMS, those are the small library size, so we do like 10 to 15,000 compounds. We already have 78 targeted proteins, and we are working on a large ESMS screen that will be almost 500,000. For the HitGen DEL, it's a library of 4 billion. We have the full definition except the smiles, but we have all the fingerprints. For the Baylor, it's 400 million. It's a much smaller library. And for XChem, we actually don't know the actual size of the library. It's probably around 8 billions, but we don't have the library definition. But every time they give us positive and negative, we have the full fingerprints for those. So if you look at the machine learning and AI models on the platform now, you click on pipeline, and you can basically choose which data set you want, which target proteins, and which model. For the moment, there is only one, but hopefully that collection will grow very soon. And then basically what you can see now, we are working on ways to look at the performance of that model in cross-validation, so when you do training and testing, and we have multiple kind of performance metrics, and this plot I think is called a spider plot. I think it looks pretty cool. And now we're going to be able to compare multiple models in the future when we have more. You can also use kind of an API, an interface, or the website itself to kind of make predictions using that model. So if you're interested in that target protein, you can use that model, put your fingerprints of interest, and generate the likelihood of binding according to this model. All the models and all the data are completely open source on the platform. So here you actually have the version code of that model with the tutorial on how to use it. And finally, on the platform, we try to provide as much documentation as possible. And the idea, obviously, is that if you have somebody like me who's not a chemist, but knows a little bit about machine learning and AI, see all those data sets, understand the mission, but have never really played with those data before. We want to make sure that they have all the documentation about the technologies, the data format, the data dictionaries, so that any kind of ML, AI experts could kind of learn the process of hit discovery and how to contribute to it. So with that, I would like to thank the AIRCHECK Dream Team. You see Nabin here and Shay in the group, and I know Mathieu and Max are in the room as well, so big thank. It was a lot of work to pull that off, but very, very pleased with the results so far. And with that, I thank you for your attention. There is a conference in Toronto tomorrow and the day after on AIRCHECK. We kind of time it with the Conscience workshop. And there is also AACR Spatial AI conference with a session on hit discovery in Montreal in July 10-12 if you're available. And with that, I thank you for your attention.
Our next presenter is Oren Kraus. And Oren and I have been crossing paths now for a little while in various things. We've been involved in biotech in the Greater Toronto, Greater Canadian setting for quite some time now. Oren is currently the Associate Director of Machine Learning at Recursion in Toronto. Many of you might know him, though, also as a co-founder of Phenomic, which was a very interesting machine learning company out of Toronto for the last little while. But Oren has a long history of being involved in machine learning and basically large data sets and using it for biotech and drug discovery for quite some time. But at Recursion right now, they've been making some sizable data sets available through their RxRx platform, and I will leave it to you to talk about it.
Great. Thanks, Owen. Great. So good afternoon, everyone. Like Owen was saying, today I'll talk about the imaging data sets that Recursion's released. So unlike a lot of the talks today, Recursion focuses on phenotypic drug discovery. So instead of optimizing compounds directly for a target, like most pipelines work, we focus on cell-based images, and we can use this in multiple different contexts. And the idea is that we can figure out, without exactly knowing what the mechanism is, what biology is being perturbed by different genetic or chemical knockouts. So this is kind of the historic context for Recursion. Over the last few years, we've kind of grown into a lot of other data modalities. So we work in omics now. Phenomics is still a big bulk of the platform. There's also a lot more chemistry modeling happening. We have a vivarium, so we even collect a lot of in vivo data from our programs. And now that we're a clinical company, we have data from clinical trials as well that we're starting to apply data science techniques to. But the talk today is still going to focus on phenomics because that's for like the data sets we've released. They're mostly in this space. So how does phenotypic drug discovery work? This is a classic example where you can build a disease model. So whether it's a gene knockout that's associated with a disease or even a virus or a bacteria that you can add to the cell culture, you generate a disease model, like a cell-based disease model. And then you still have cells that are untreated. So those would be your healthy controls. And essentially you want to find compounds or other modalities that will kind of revert the phenotype and make the cells look more like the healthy control. So this becomes a really interesting machine learning problem because you need to figure out how to featurize those images in a way that you can relate them to each other. And you also need to account for all the different kind of experimental artifacts. So typically if you repeat the experiment, the cells can just look a lot different from experimental batch to experimental batch. So this is a visualization of what that can look like in practice. So this is just a 2D plot showing the disease condition, which is kind of the starting point. And then if we add different compounds at increasing concentrations, we can kind of score how well they're doing at moving the phenotype back to the healthy state. So in terms of the data sets we released in this space, the initial one was released in 2019 called RxRx1. Since then, we've released 5 additional ones. 3 of them, which I won't go into much detail on, are related to COVID-19. So that was Recursion's way to contribute to the broader community by screening cells exposed to the virus and the different kind of treatments that were being considered at the time. And then the 2 more recent ones are RxRx3 and RxRx3 Core. So I'll go into more detail into those ones. In terms of stats that I was able to collect on these, so RxRx1 was specifically affiliated with a Kaggle competition. So there were about 800 teams and 1,000 like individual users contributing to that in 2019. So that really like spurred a lot of interest in cell imaging data sets in the machine learning community. RxRx3 is a massive data set. So compressed, it's about 83,000 gigabytes. Uncompressed, it's over 100 terabytes. And it was released about 2 years ago. So there's a bit of a barrier to downloading it, but it's still been downloaded over 6,000 times. And RxRx3 Core is a subset of RxRx3 that was just released last November, and it already has about 1,700 downloads. So these datasets have contributed to a lot of publications, like both internally and externally. So overall, they've been cited over 160 times since 2019. And if you aggregate all the datasets together, they've been downloaded over 9,000 times. So yeah, going into detail on RxRx1. This is the imaging format that we use at Recursion in general, but was used in the dataset as well. So it's a modification of a popular assay called cell painting, where you can stain different parts of the cell morphology with distinct markers. So that gives you kind of an overall view of the cell structure and cell compartments. And then those, in our case, it's a 6-channel image. And you can see the composite version of that on the left-hand side. So you typically will apply different perturbations on the same imaging protocol that's stayed consistent over, more or less since the beginning of Recursion. So, we have, 5+ years of data in this format that can all be related to each other. Whereas in a lot of industry use cases, like phenotypic screening is used very much for specific programs. So the assay or like the experiment itself is kind of customized for a specific protein or cell type, and it's kind of hard to relate data across those experiments. The main challenge we were trying to solve with this data set is improving batch correction or batch alignment. So in this case, you can see 2 different siRNA perturbations in each row, and then what those same images look like across different experimental batches. So what's striking is that the difference between the experimental batches is a lot more obvious than the difference between the actual treatments. But that's something we all want to filter out when we're doing this kind of analysis. So the way we designed this data set, it had 1,100 siRNA treatments, and then it was repeated, the exact same design was repeated across 51 experimental batches. It also included multiple cell types, so that's the distribution of cell types that were used and the number of experiments that had those specific cell types. HEVIC was the bulk of it, which is the cell type we used in most of our experiments. And it was set up as a classification challenge, so teams had to train a model to predict what the treatment was, like out of those 1,100 siRNAs. And then there was a held out private test set that had batches that weren't included, experimental batches that weren't included in the training set. So overall, there were 34,000 images that had to be predicted that weren't part of the training set. Because this was intended as a batch alignment challenge, we actually included the controls from the test set because we thought that would, we thought the teams would kind of leverage that in their modeling. That didn't exactly work out. They just got to use that to cheat, but it's okay. So this was, like I was mentioning, a Kaggle competition in 2019, and the top performing team or individual achieved 99.763% accuracy. So overall, that means that they only misclassified 36 out of the 15,000 test wells, which is super impressive. That individual actually joined Recursion a bit after the competition, and he's been working with us since. So that's one benefit of doing these kind of challenges. But it also spurred a lot of innovation at Recursion because that kind of got the company switching to leveraging deep learning models across the platform. So how do we use these models? I was kind of explaining before. This kind of plugs into the pipeline for scoring phenotypic screens. So essentially, even though the model's trained as a classifier, you can use the last layer or like a few layers before that as a feature vector. So you can take an image, and instead of having this complicated unstructured data set that's like pixels by like XY coordinates, now we can just have like a single, in this case, 128 dimensional vector that represents like that image crop. So with that, after some alignment and normalization techniques, we can use pretty simple distance metrics like cosine similarity to define if two treatments are similar to each other. So for the brute force screening approach, basically we see for a treatment how much of the direction of that treatment is kind of opposing the disease model and pushing that feature vector back toward the origin, the healthy control. In some cases, we can even do phenotypic-based structure-activity relationships. So you can even see, based on the similarity between the perturbation and the healthy control, you can see that with increasing doses, we can actually define a disease window for compounds based on phenotypic data, which is pretty unique. An example of where this has been successful is in the COVID screening I was mentioning before. So in response to COVID-19, Recursion screened 2 different cell types with the virus across a bunch of the compounds that were being considered in clinical trials. So in this plot, you can see an example of remdesivir actually having kind of the desired effect where the increasing doses are moving more toward the healthy control, but other compounds that were considered at the time, like hydroxychloroquine, actually has some pretty bad off-target effects. So it seems like it's improving, but then it actually gets closer to other phenotypes and away from the healthy phenotype that we'd want. So at the time, we kind of scored a bunch of the compounds that were being considered, and out of the 7 that we evaluated, we predicted 6 of them correctly, whether they'd be approved in randomized clinical trials. So it's like pretty impressive demonstration of how the platform can be used in drug discovery. But beyond the brute force screening, like there's a bit of a challenge there because any program we want to work toward, we need to define the disease model, which can be challenging. Like not every disease is as simple as knocking out a single gene and then imaging it in a well. Also, you have to screen your whole library every time you want to do that, so that's not very scalable. So a capability that actually arose over the years was this ability to infer biological relationships just by looking back at all our existing data sets. So here's an example of how that works. If we look at a classical biological pathway, so in this case it's the mTOR pathway. TSC1 and 2 inhibit mTOR, so it's kind of well known in literature. And if we look at the phenomic relationships I was mentioning before, based on the cosine similarities, we can see that TSC1 and TSC2, are relatively close together, but then mTOR kind of has this opposite effect. So you can imagine that can be a very powerful way to just understand which genes are related to each other, and also even if we start to add compounds, like seeing how which compounds might be good at inhibiting or being similar to the knockout of a gene. So this is kind of the way Recursion uses most of our phenomics data these days. We call it a map of biology. But to improve on the modeling, we also had to develop benchmarks for this. So this is based on a paper that was in PLOS Comp Bio last year. And the way it works is we have genome-wide screens where we knock out every single gene in the same cell painting assay. And then we curate annotated relationships from known databases like Quorum and StringDB and a handful of other ones. And then we look at the most similar and most opposite relationships across the whole data set that we've generated. And we compute the recall of the annotated relationships at those extremities. So if you see like the bottom right corner, there's an example where in the green box, those are true positive, so relationships we're capturing, and then the red box is relationships that we're missing that are annotated. We don't want to penalize the model for making predictions that aren't annotated because they could have just never been evaluated. So that's why we only focus on recall. So using this as a benchmark, we've kind of stepped into seeing where we can go with the modeling because the initial Calgo competition used a technique called supervised learning and then transferring that model onto other data sets, which initially came out of papers like AlexNet that really drove the field, the computer vision field a lot in the early 2000s. But now, a lot of the machine learning communities shifted to scaling models, kind of like in the language model space. So the way you do that is you grow your data set, you grow your model, and then you throw a lot of compute at it. So those two plots, then right behind me, yeah, those show that the growth in model and data set size is particularly since 2020 has been pretty explosive. So we've seen this with LLMs and with vision models. Along with that, the growth in compute's been pretty substantial as well. So that'll memes from Richard Sutton's blog post about, instead of trying to think too hard about the math and like the statistics, just, throw more data, throw more compute and use like an unbiased model like a transformer. So as these larger data sets aren't really annotated with labels, the underlying technique, the way most of them are pre-trained is with self-supervised learning. So there's no actual annotations, you just take the raw data and for the most part, the models work by masking out part of the input and trying to predict it. So with language, it's kind of next word or next token prediction. There's a version of this for images called masked autoencoders that's also become really popular since 2021. In which case you mask out like chunks of the image, the transformer gets an embedding of that and then tries to predict all the missing patches. And if you train that over very large data sets, you can actually get a pretty good performing model. And that's where RxRx3 comes in. So I mentioned before that it's the largest data set we've released, like over 100 terabytes when it's uncompressed. It includes a genome-wide knockout, so all the CRISPR knockout of like 17,000 genes. It includes 1,600 compounds, so the majority of the ones that are FDA approved. And it's still the same 6-channel image format I was mentioning before. So overall, it contains over 2 million wells, and it's been downloaded over 6,000 times. So leveraging a data set like this, myself and my team were able to train one of these large-scale vision transformer models. So the way this works, like I was mentioning before, you mask out the image, you try to predict the missing patches, and then when you actually want to use the features from that model, you just take like the intermediate layer before the model's trying to decode the missing patches. So we showed in a paper that was in CVPR last year that as we increase the data set size and the model size, we continue to see improved performance on that recall metric. So it kind of showed that the scaling hypothesis also applies to microscopy data sets. So despite this data set being a great resource for training these kind of foundation models, we actually found that it was a bit challenging for a lot of researchers to use because not a lot of researchers have 100 terabytes lying around that they can just download this data set for. it also didn't really come associated with any benchmarking tasks. So if you're just trying to train machine learning models on it, it wasn't really clear what to do with it besides like using it for training data. So that's where we released RxRx 3 in November last year. So it's a subset of the full RxRx 3 dataset. It's only 18 gigabytes in size, so you don't even have to download it. You can just stream it directly from the sites where it's hosted. And then we also released it along with zero-shot drug-target interaction benchmark. So yeah, this is the overall stats. So this is a similar image to before, but the main difference is that now we only have 736 genetic knockouts in this data set. And then instead of providing the full 2048 by 2048 image, we just provided the center crop. So that reduces the data set quite a bit. And instead of 2 million wells, it only includes about 10% of that, so 200,000. We also applied a few other tricks to get the data set to be a smaller size. So we converted it to JPEG 2000, which was also like a 16-factor compression, and then used uint 8 instead of uint 16. So all these tricks like don't really reduce the quality of the data, but make it much more easy to download and use. In terms of the benchmark, so this is the visualization of the drug target interaction benchmark. So we have the 16,000 compounds in the data set. And basically you rank the similarity of those 16,000 compounds to all the genes that are unblinded in the data set. And we compute average precision to see like where the right target scores for every single compound. And we do that across the different doses. So here again, you're seeing that the larger vision transformer models are outperforming the smaller ones, and as well as the like cell profiler and a random baseline. To encourage the community to use this, we released it both on Hugging Face, so that's just literally 2 lines of code to get that loaded and start using it for training or evaluation, and on Polaris, which my colleague Cass talked about yesterday. So that's the Polaris Hub Bivalence. So it's another way to access the data, and I think the benchmarks are even hosted on there too. So that's just like a one-stop shop for using and benchmarking models on these data sets. So this is a bit off topic, like the topic was mostly focused on the data sets, but I can mention a bit about how we've used them. So in addition to that model I talked about that was in CVPR, we kind of scaled this up even further last summer. So the first model we named Phenom 1. Phenom 2 was presented at a NeurIPS workshop in December. So here we made the model 6 times bigger, so now it has close to 2 billion parameters. We actually made the data set smaller, so we found that like curating the data set and leaving out a bunch of controls that were pretty similar to each other led to better training. But we put like 4x the amount of compute in it, so it took like about 256 GPUs training in parallel for a week to get this model trained. But we continue to see like important performance improvements even with that model. So the evaluations shown here are on an external data set. So this is by the Jump Cell Painting Consortium. So that was another open data science effort in this space led by Ann Carpenter of the Broad Institute to also screen a lot of genetic knockouts and compounds in the cell painting assay. So here you can see that pretty much all the vision transformer models are outperforming cell profiler, which is kind of the classic feature extraction segmentation type software that the community uses. And again, you can see that the larger models are performing better than the smaller ones. And finally, we don't just release the data sets, we've also released one of these models. So it's called Open Phenom. It's also available on Hugging Face and maybe Polaris. So this one's a channel agnostic model, so that can actually, it doesn't really matter which fluorescent markers you use. Like a data set might only have one channel or even a bright field channel. You can still use this model. It was all trained on all open data sets, so the RxRx 3, which is open source, and like the JumpCP data, which is also open source. And this is probably like our most impressive download number, but since November, it's been downloaded 50,000 times. So I'm excited about that. It shows that it's being used for lots of different applications by lots of different teams. And yeah, I'll end there. So thanks for listening.